It's my pleasure to moderate this session. I'm Susan Halabi. I'm a professor at Duke University and a PI with the new funded CERSI, the Duke UNC. So as you all know, following the 21st Century Cure Act in the US, real-world evidence has been well defined. According to the US Food and Drug Administration, real-world data are data, in quote, so this is verbatim, data relating to patient health status and or delivery of healthcare routinely collected from a variety of sources. Real-world evidence is, in quote, clinical evidence about the usage and potential benefits of risk of a medical product derived from analysis of real-world data. RWD can be from a variety of sources, such as data derived from electronic health records, medical claims data, data from product or disease registries, and data gathered from other sources, such as digital health technologies that can inform on health status. Now, of course, with anything that's new, with the great emphasis on RWD and real-world evidence, has come a variety of challenges and novel ways to optimize and enhance patient centricity in the development of new therapeutics, whether by informing target selection, designing randomized clinical trial, or enabling personalized medicine. This includes novel ways to evaluate disease burden, patient heterogeneity, increase ability to access, generate, and leverage fit-for-purpose data sources, and opportunities to harness cloud computing for enhanced scalability and interactivity of digital tools. So in this session, we're going to hear from three great speakers. I'm going to introduce the first speaker, and I'm told not to talk a lot, so I'm going to be brief. Our first speaker is Dr. Molly Jeffery, who is a co-PI of the Yale-Mayo CERSI. She's a health economist and health service researcher who studies how people use healthcare and health insurance. Dr. Jeffery serves as the scientific director of Mayo Clinic, Division of Emergency Medicine Research, helping to design, conduct clinical trials, quality initiative, and large databases aimed at understanding and improving the care patients receive in the emergency department. The title of her talk is Understanding the Use of Existing Real-World Data for Medical Product Evaluation. Please welcome Dr. Jeffery.
Thank you so much, and I am excited to be here to present briefly on this project on behalf of a much larger team. So I'm going to keep it focused at a very high level and assume most of you are not thinking about target trial emulations and real-world data in your day-to-day life. If you are, I don't know, like maybe take a bathroom break or whatever. I'll try to not get too far down into the weeds. But just as an acknowledgement, this work was funded by the FDA through the CERSI, and the standard disclaimers apply. Our team is just a fantastic team across Yale and Mayo and other institutions now as well, and was led by Josh Wallach and Rosalina McCoy. So I know that you have all seen the evidence hierarchy. The observational studies are kind of down towards the bottom. RCTs are near the top. And systematic reviews and meta-analyses the very top right. But I think that there are lots of problems with them, and I think you probably have all thought about those problems. So up at the top, with systematic reviews and meta-analyses, garbage in, garbage out, right? So we've all seen meta-analyses that use crappy studies. We have all seen systematic reviews that conclude we couldn't answer any of our questions because there isn't enough data out there answering the questions, right? So that's a real problem. RCTs have real problems as well, and I know this is a little bit heretical if we have a lot of FDA folks in the room, but I mean, we all recognize these issues. They work really great at causal inference when they're conducted well for the people who were actually in this study. But the real world of how these drugs are used and how products are used is very different from what we see in RCTs. And then obviously, with the observational studies, we have issues of confounding and figuring out how people end up on the drugs that they have, right? So they all have problems. And I think the evidence-based medicine community has kind of started to move away from that hierarchy and started to think more about like a pie or like a circle of, I used Dali to create this. This is Dali's interpretation of horses for courses, right? So the idea is you don't use like a quarter horse to run a race. You don't have them pull a big wagon. There are horses that do that really well. But they all have great uses, right? And so every kind of data is needed. Every kind of study design is needed. We just need to make sure we're using them thoughtfully and correctly to answer the question fully. And so just to get a little bit more explicit about the pluses and minuses of RCTs and how real-world data can kind of work together with RCTs to answer questions, that big challenge with RCTs is generalizability, both the setting and the participants. So we don't see a lot of minoritized folks. We don't see a lot of people from rural areas. We systematically exclude women of childbearing age, particularly older folks. And that's a real problem, right? And then settings, they're all conducted in places that have a lot of resources. Academic medical centers that don't look like the rest of the world, where the majority of people get their care. And I actually just looked this up, and 85% of hospitalizations in a Medicare population are outside academic medical centers, right? So that's not the real world. And real-world data's advantage is that it captures a much broader segment of the people who are out there and looks at the drugs and devices as they are actually used. The caveat here, obviously, is that we don't have a public health information system in this country. And so we depend on different types of data sources, like claims data, EHR data. They all have problems. They're all missing people as well, but they look more like the real world. In terms of cost and time, I know that you guys all know this. A phase 3 trial on average is $20 million, and it took millions of dollars to even get to the phase 3. They go on average for 40 months. In comparison, this study that I'm going to tell you a little bit about went for about 2 years and cost about $250,000 to emulate 2 trials. Still very expensive, right? But one of the really terrific things about real-world data analysis is that there's low or no marginal cost for additional follow-up time and for additional participants, right? So you get as many participants as you can get without paying more money for that, but still expensive. And then in terms of priorities, RCTs are going to be just where the money is. And so it's going to tend to be on newer drugs and more expensive therapies. And the existing and less expensive ones and the ones that can make a difference across a huge swath of the population are going to be a lot harder to fund. Real-world data is easier to fund. It's got a lower cost, so it just has a broader remit. But the caveat here, again, is that the least expensive stuff, so if you think about claims data, like over-the-counter drugs, not included. Any of the dollar prescriptions at Walmart, not included, because people are just going to pay their dollar and not go through insurance. So still caveats. But if we're going to use real-world data, we need to be very thoughtful about how we do it. And this paper came out in, I think, 2016, maybe published online in 2015, and has just been a real thought-provoking and influential paper that said, if we don't have an RCT, if it's not feasible or it just doesn't exist, how should we think about analyzing existing data to emulate a target trial? So the idea is, instead of just analyzing our data as an observational and maybe comparing like current users to never users, we think hard about how an RCT would work, and we design our observational analysis to replicate that as much as we can. And so, one thing would be, instead of current versus never users, you only look at new users versus never users so that your time zero is the same for everybody, as an example. And I think the work that I'm talking about today builds on that a little bit. So what that was talking about is really looking at observational data with an RCT lens. So how do I design my observational study to avoid certain biases? And what we did in this study is look at observational data and RCT data together. So I've done my target trial emulation. How well did I do at avoiding some of the biases I know are going to be present in observational data? Did we actually do a reasonably good job when we compare the results together? So this is the study. And there's a couple of things that were interesting about what we did. But one of the things that was particularly different is we were looking at ongoing RCTs. So you think about all the different ways that observational data can be biased. There's lots of them. But if you think about a target trial emulation, if I pick a trial that's done, it's been published, and I know the answer, now I've brought in the bias of researcher degrees of freedom, right? So I can design my analysis, and if I know what answer I'm supposed to get, I might subconsciously start to tweak the way I'm doing my analysis to start to get at what I think the answer is, instead of going at it from, sort of that sort of pure standpoint of saying, what is the right way to do this? And so we picked two ongoing trials that had not been published. We published before they came out. And you can see we've got 3 publications on this. There's one for each replication, and there's one that compares the replications to the published trials, and so you can go look those up to see more of the details. But these are the 2 trials. GRADE was glycemia reduction approaches in diabetes, so it was people on metformin with relatively new diabetes who had another drug added, and then PRONOUNCE, which looked at whether, I'm terrible at pronouncing these, but I think it's daguerreolex, had better cardiovascular outcomes than leuprolide, and there was a reason that you would think that would be the case, and they wanted to see if that was actually the case. The first question is whether you can actually do it in observational data, right? So feasibility. We looked at the studies. So we chose these studies thinking that we would be able to do a very good job of capturing the right data points and measures to make this a worthwhile endeavor. But we did still look at it and say, okay, of all the measures that are reported or called out in the protocols, which ones can we do well versus not very well with observational data? And it was about a third. So about a third were very readily ascertainable. Patient age is pretty obvious. You can usually get a measure of sex. Recent procedures, many medications, most comorbidities. Partially ascertainable means either not everybody had the data or we could get like a proxy for it, but not exact. So an example would be smoking status. We think codes for smoking status, current smoking status, are probably pretty good, but we don't really have like history of smoking very well. In this particular data set, we were able to get HbA1c lab test result, not just whether it was done, but the actual result because of the data set that we used. But you can't always do that. And we certainly couldn't capture it at the same cadence as it was measured in the study, right? So they did it every 3 months. We just had whatever was actually done. And then not ascertainable at all. You can't get a disease stage or score for the most part. You can't get people's weight or BMI. Like I said, over-the-counter drug use, most lab tests, imaging test results, things like that. So we cannot capture them, but that doesn't mean we can't do this target trial emulation. We just have to be thoughtful. So I'm not gonna belabor this, but this is just a comparison of the population. So this is on the left is the emulated trial. On the right is the RCT. You can see we had a much larger sample size. So we had 1,100 in each arm. They had to stop recruiting early. They aimed at 900 and stopped at 544. Our group is older, which is good because it's more like the disease population. And then we actually had substantially more African-American participants in ours than they did. So some differences, where we might actually see differences in the results when we compare them, but also a really important measure of looking more like the real world. And then in terms of grade, similar things. So again, older, more like the disease population, and we had many more women than were in the trial. So this is just like a scorecard of how do we do in replicating the RCT. And this is kind of like how this is done. And we've got these 3 different ways of measuring concordance between the emulation and the trial. So did the confidence intervals overlap between the RCT and our result? Were they the same direction of effect? So if daguerreolex is better than leuprolide in the RCT, was it also better in our study? And then this, I don't like the statistical significance one, because it's going to be a function of how many people you have, and we had like 4 times as many people, right? But the question is, were they both statistically significant at 0.05, 0.01, 0.001? And you can see some interesting things. So one thing that you can see is we did really well at direction of effect. The only one that did not work out that way was all-cause death in the pronounced study, and we can talk about why that might be. And they had in their protocol that they were going to do angina, but they did not actually report it, so we were not able to compare. Oh, and one thing that I didn't say, which is interesting about grade, is that there were 4 arms in the original trial. We had to drop one. We had to drop the insulin one because, I think it's insulin glargine. We had to drop it because that group just did not look like the rest of the population. You can think of lots of reasons why that would be, right, in a type 2 diabetes population. So we actually just had to drop that group. So that's why they don't show up here. So I think really exciting. I think the results of this are really exciting, really interesting, especially given that we did not have that researcher's degrees of freedom, right? We were out before they were published, so we didn't know what the right answer was going to be. The caveat to target trial emulation and all observational data is we do not have randomization, right? So we had to drop insulin glargine because that group just looks too different when you look at it in the real world. It was just too different. And then limited measures, right? So there are certain things that we just cannot capture well in real-world data, at least in the data sets that we have. If you think about lab tests, in almost all claims data populations, you can't get lab test results. You could say, well, let's use EHR data instead to do these things. But if you've seen one healthcare system, you've seen one healthcare system. So you can try to figure out ways to pool data across healthcare systems and things like that, but it's very, very hard, and there's not a lot of incentives to do that. So it's just very tricky to think about. And I always have caveats to my caveats, right? So here's the caveats to those caveats, right? Well, there is a lot of random practice variation in this country, right? If you look at the Dartmouth Atlas, people do things very differently. We can think really hard about how to design these target trial emulation analyses to take advantage of some of those things. And can we add more observable factors, right? So I don't know if you know this, but Medicare does not actually ask people what their race ethnicity is. When you use a Medicare data, there is a measure there that is an imputed measure, but they're going to start asking, right? So CMS is starting to try to figure out how they can start asking people that. So we can start to collect more data that we know is important for the purposes of research and for the purposes, right? So what is claims data for? Claims data is for getting paid. But if we can put some incentives in place to kind of say, well, we need to collect a little bit more information than just what we need to get paid, and put some teeth around that, we can start to improve this kind of data really well. And then in terms of limited measures, let's think about linking data sets. So it's really hard to link EHR and claims data, but let's think about how we can make that easier. Stacy Doucet-Zena at Vanderbilt has done it. It was incredibly hard, but somebody's done it now. Can we learn from that? Can we start to think about how we can build bigger sets of EHR data that are meaningful and readily usable? And so that was kind of my 30,000-foot trip through target trial emulation. It's something we're really excited about and continuing to work on. I think the next steps with this idea of considering RCTs and real-world data together is now to say, okay, well, we do a pretty good job of emulating in these particular trials. Now, what can we learn from our real-world data that we couldn't learn from the RCTs? What kinds of subgroup analyses can we do? What kinds of other things can we do to build on the fact that we know we did a pretty good job of emulating? So now let's start to think about what we can do next that couldn't be done with just the RCT. So thank you all.
Thank you very much, Molly. We're going to hold questions until the end of the session, so stay tuned. I guess our next speaker is online. Our next speaker is Dr. Carter Cow. Is he online?
Yes.
Oh, thank you. Hey.
Hey.
Dr. Carter Cow is a associate professor at UNC Eshelman School of Pharmacy. He has been working a lot in the field of pharmacotherapy and experimental therapeutics, and he has done tremendous work and developed expertise in numerous areas. Please welcome him for the next talk.
All right. So let me start by saying what a wonderful symposium. This is my first time taking lots of notes. Thank you for all these organizers and the speakers. And thank you for the invitation as well. Truly a great pleasure. Today, I'm going to talk about a project has just been founded last year. So there won't be much of progress report in this presentation, though. I'm going to stay on the high level, focus on the backgrounds as well as the motivation and the challenge we are facing in doing this project. Here's the outline. So historically, in developing oncology drug, I think MTD is the most prevalent approach, right? At the early phase of clinical trial, we challenge the patient step by step. We call this a dose escalation study until we reach a point the patient cannot tolerate to the highest degree or the maximal degree. We call this a maximal tolerate dose. So we typically select this dose, move on to do a comparison with the current standard of care so that we can get the drug, show whether it's better or worse than the standard of care. This type of approach, MTD, have a underlying assumption, which is that the more the better. The more dose, the better of the efficacy. That's the fundamental assumption about this approach. Actually, this assumption rooted in the pharmacology of chemotherapy to a large degree. If you look at the two panels here, chemotherapy, drawing the dose dependency of the chemotherapy in terms of their toxicity and the efficacy, those two curves go hand in hand. You're not quite separable between those two curves. That's pretty much the reason why we choose the highest dose, the most patient can tolerate, so that we can do the comparison study. However, this not always be the case for the molecular target therapy. On the right panel, you can see for molecular target therapy, which has been have been dominating the approvals in the last 5 or 10 years. For the target therapy under the MTD paradigm, you may potentially select a dose way higher than the dose can saturate the efficacy, leading to unnecessary toxicity. That's the problem. And also for immunotherapy, we all know that the efficacy and toxicity are largely depend on the tumor biology as well as the patient host immune system. If you do a dose dependency analysis, you can often observe this flat relationship. That's also tell us that MTD approach probably is not applicable. If you you can identify a maximal tolerant dose for the immunotherapy, sometimes you cannot. You're probably already way beyond the point necessary for efficacy. You can put the patient in danger. So how to deal with that? I think FDA has been well aware of this issue for many, many years. If you look at the workshop being organized by FDA in the past 5 or 10 years, they focusing on this dose selection topic a number of times with ACR, ASCO, ISOP. There's lots of discussion around this topic. Several years ago, FDA started this initiative called Project Optimus Initiative. The whole thing about this initiative is challenge the MTD approach, try to encourage the sponsor to think about dose optimization studies. Right after they select the maximum tolerant dose, the FDA kind of encouraged the sponsor to think about a number of doses to compare them, try to maximize the benefit and risk ratio. That's probably a dose being selected lower than MTD and that dose may give you the most benefit and risk ratio. That's pretty much the current thinking about Project Optimus. This initiative started several years ago. Let me give you an example, why FDA started this and what's the real world situation about this challenge. So PI3K inhibitor, this is a category of drugs. PI3K actually is a group of enzyme inside the cell responsible for cell survival, proliferation, differentiation, as well as motility. It's a natural target for cancer treatment. There are a number of drugs has been approved. If you can see my mouse here, the number of drugs has been approved under the accelerated approval pathway by FDA for hematological tumors, a number of hematological tumors. Majority of these trials based on the overall response rate, ORR. Please note that this type of drug, the clinical trial started after, or not after, before the Project Optimus initiative. So there's a very limited dose exploration. So majority of the sponsors kind of select the dose to the high end. I'm not saying maximum tolerate, but it's pretty much the dose the patient can tolerate to a large degree. There's a very limited dose exploration at the early stage of these trials. However, when the sponsor moving on these trials in the confirmation stage, lots of patients suffer from high toxicity. Some of this toxicity not even bearable by the patient, leading to a loss of dropouts and the discontinuation of this treatment. Some of the toxicity even associated with the worst patient survival, showing a potential detrimental effect to the patient survival. So let's zoom in about one particular PI3K inhibitor, adilisib. You can see the adilisib arm colored in blue and the control arm. This is a bar plot summarizing adilisib RCT trial results in terms of the toxicity. There are a number of indications we're talking about. The blue color is the bar represents the target therapy, and the green color is the control arm. You can see the adilisib in general have a much higher fraction of patients suffer from grade 3 or even higher toxicity. Majority of the toxicity are related to severe toxicity. Because of that, and many patients discontinue the treatment. There are about 40% of the patients discontinue the treatment because many of these toxicity cannot bearable. And some of the patients even reduce their dose and interrupt the dose. The event happens throughout the trial is substantial. Those concurrent events drastically compromise our interpretation of the clinical trial results. If you want to learn more about this type of drug, safety and efficacy profile, you can go to the website FDA posted. There's the ODAC meeting 2022, nicely summarized the challenge around this type of drugs. There's a lot of debate between the sponsors and the FDA on this topic. But in general, you can see that the dose being selected on the high end, sometimes the patient going to face a high challenge to take these drugs in a long term, and also some of the toxicity associated with this type of the drug can lead to unnecessary detrimental effect to the patient's survival. This is not only for the PI3K inhibitor. If you look at the whole landscape of target therapy, majority of them are tyrosine kinase inhibitor. There are more than 50%, for some of the target therapy, more than 50% of the patients have to go to a dose interruption or reduction so that they can tolerate. You can see that this is not only happened for PI3K inhibitors. Majority of them are tyrosine kinase inhibitors, including the EGFR targeting antibody. This is something we also observe in the real world situation. It happens very frequently and broadly. So the question back to the real world evidence, how can we use the real world evidence data or real world data to help us to address some of the challenge? I give you a very brief background about this real world data. So FDA, if you look at the website, there's a number of documents highlighting the guidance about using the real world data to support our regulation and drug evaluation. The number of things we can do using the real world data. One is the evaluation of the treatment effectiveness. And also we can use that to evaluate the patient tolerability and the patient compliance. This is kind of related to the aim we try to address. And also, if you go further, you can understand what's the confounding factors make potentially related beyond the RCT data. And there's a heterogeneous treatment effect, HTE. Use that prognostic and predict factors, we can better understand the conditional average treatment effects, and so on and so on. You can use the real world data to help us understand the disease trajectory, and maybe create or synthetic some virtual control arm for us to do some evaluation in a situation, the rare disease situation, et cetera, et cetera. And the sources of the real world data are multiple fold. The data we are evaluating largely come from the electronic health record, EHR, as well as the pharmacy records. But beyond that, there's data being mentioned in the previous slide, in the previous presentation. They relate to the medical claim billing data, as well as the disease registry. There are many sources of the real world data. So what's the type of question we are interested? I listed here that we know that in the real world patients, particularly in our UNC healthcare system, there are many, many patients cannot bear the dose being prescribed or being labeled. They modify their dose. When I say dose modification, they are threefold, including the discontinuation of the treatment or the dose reduction or the interruption. There are threefold of dose modification. We try to evaluate whether the dose modification due to the treatment intolerance can compromise the clinical outcome or not, whether that's going to influence the patient's survival. If the answer is no, then we kind of think this dose being labeled maybe are too high. We should think about a lower dose. If the answer is yes, we don't know that yet. If the answer is yes, then we probably think about alternative therapy or think about some strategy to mitigate this toxicity or side effects. But this question is not that straightforward, because you can see that those modifications also closely associate with patient toxicity, adverse effects. There's a second layer of the question is, which is, the second layer of question is whether the treatment associated toxicity has any direct detrimental impact on the clinical outcome, right? The dose modification closely associated with toxicity. We don't know whether the toxicity or the dose modification, which one has a stronger impact on the clinical outcome. We should think about some of the advantages, statistical approach, try to figure out which one is the reason, which one is the association. To give you a high level summary, what the data come from. Within our UNC healthcare system, there are many hospitals associated with this system. We can to pull out the electronic health records, mining the relationship between the dose and toxicity, as well as the dose efficacy. That's pretty much what we try to do. We are in the process of pulling out the data now. One thing I want to point out is that within the system, there are about half, 50% or 45% of the patient have their pharmacy records or pharmacy refilling records being kept within the system. That's the unique advantage of our analysis, because we focus on the dose aspects of this efficacy and toxicity analysis. We want to know exactly what's the pharmacy refilling records and history for each individual patient. Using that within our system, we can match up on these pharmacy records and the patient, patient safety and efficacy profile on the individual patient base. That's the unique features of this data set. So what's the challenge we are facing? I think, if you think about the tumor or oncology drug development, patient or tumor heterogeneity is, if not the most challenging aspect, probably is one of the most challenging aspects. This heterogeneity across the individuals give a lot of trouble to think about the control. Most time we want to compare the patients with dose modification, without dose modification. It's not straightforward, because those group of patients differ in many different aspects. Sometimes you end up with an inadequate control. You should think about how to balance these two arms when we try to compare or evaluate the dose dependency. So that's a challenge we are facing in analyzing the data we collect. Another thing is the population average cannot represent, fair fractions or individual. That's pretty much the same point as the first one. If you have a very heterogeneous distribution of patients, the population average probably only represents a small fraction of the individual patient. Majority of the individual patient cannot be represented by the population average. There's a challenge to derive the individual response and safety profile. That's pretty much the things we try to using some advanced statistical approach to get there. Again, to give you a quick simulation example to show what I mean about the patient heterogeneity. This is a dose response curve we pull out from a drug being approved by FDA without naming the name. You can see that there's dose dependency on the x-axis, that's the dose and response on the population level. From the population level, it's not that hard to tell that 90 units of the dose is probably better than 40 because they can elicit a higher response. However, if you do a simulation on the individual level, we call this an ICE simulation, individual conditional expectation. The approach of doing this is just fix all those covariates on the individual patient level, simulate individual dose-response relationship. You can see there's a huge heterogeneity across individual patients. If you pick this group of patients under the 40 dose units, you can see the ECOG equal 1. I think some of these previous presenters explained this ECOG. ECOG is essentially related to the patient physical activity. ECOG equals 0 is kind of indicating the patient have no restriction of physical activities. So if you think about this group of patients with the ECOG equals 0, they probably already have a saturated response under the dose of 40. They do not have to go to 90. We are not questioning 90 as a better choice on the population level. The things we are thinking is, if you want to compare 40 and 90, you better control the ECOG, those covariates, one of the important covariates. Otherwise, if you choose a group of patients here, group of patients on the right side, you may end up choosing 40 as the better dose options. So there's no secret that in analyzing the clinical trial data, particularly the real world data, there's many correlations, right? There's many correlations. However, those correlations cannot be interpreted as causality. In the real world data mining, we find out one of the challenge we believe is finding and correcting those confounding factors. That's essentially important for us to do either the emulation or either the dose, different group of comparison. We want to understanding those confounders to a higher degree, to a quantitative degree, so that we can better understanding the dose dependency. That's the key challenging point we are facing. So if we want to evaluate the dose dependency, we better using those confounder factors to help us to derive the causal DR relationship, dose response relationship. That's more clean. That's more representative of the true dependency on the dose. So what are those confounding factors? If you look at the whole cascade of drug pharmacological action from the dose all the way to response, you can see there are many, many factors may potentially influence the dose response relationship, including the patient demographic information, the tumor biology, the genetic backgrounds, as well as the situation related to the tumor stage, therapeutic line, ECOG, fragility. All those factors are all potentially influence those response relationship. If you categorize those many factors into a high level, so degrade, these factors can generally classify into two groups. One is the risk factors. That's a prognostic factors. The factor fundamentally associated with the disease can be potentially translatable or scalable across treatment within the same indication, same stage of cancer patients. On the other side is the treatment factors, predictive factors. They are also important for the response and toxicity. However, those factors are treatment specific, not disease specific. We should be careful scaling those factors when we facing multiple sources of data, when the patients are taking multiple type of treatments. So we try to spend some effort, try to understand the differentiation between these risk factors, the prognostic versus the predictive. So another figure demonstrates how these two type of factors can to influence the hazards of the patients. The hazards, when I'm saying that, including both the efficacy and toxicity, you can see that the prognostic factor, that's the intrinsic disease risk factor. Without this factor, both the treatment and control arm, they've been influenced in terms of their hazard ratio, in terms of their hazard values. However, the predictive factors drastically influence the experimental arm, not the standard arm. We should spend some effort. We are planning to spend some effort, try to differentiate these two type of factors so that we can better understand what type of factor we can scale across trials, which factors we cannot. This is a huge challenge. Even though we have a plan to do that, I'm not quite sure whether we can get there with the data we have. So I can to make a quick summary given the time. So Project Optimus is the initiative started by FDA several years ago. The whole thing is challenge the MTD approach or paradigm. We should start questioning more is not always better. And there's a lot of patients modify their dose due to intolerability. And this dose modification not only happens for PI3K inhibitors, can happens across the board within many type of molecular target agents. We believe one of the challenge in our analysis, at least, of mining those real world data is finding those confounders and then incorporate those confounders to help us to correct those influence of the confounders so that we can have a more cleaner dose response relationship. And some of the confounder can be learned by using the historical RCT trial. We already have some of the RCT trial data requested from some of the platform, as well as using the real world data we collect either from our own system, or we also are planning to collaborate with some national institute to get some more broad information around those target therapies. So I'm going to stop here. Huge thanks to all those collaborators, including the collaborator within UNC as well as FDA. Thank you so much.
Thank you very much, Carter. Our third and last speaker for today's session is Dr. Danya Qato. She's an associate professor in the Department of Practice Sciences and Health Outcomes Research at the University of Maryland School of Pharmacy. She is also a practicing pharmacist, epidemiologist, and health service researcher with a PhD in health and pharmaceutical service from Brown, a doctor of pharmacy from University of Illinois, and masters of public health with concentration in international health from Harvard University. The title of her talk is Epidemiological Challenges to Using Real-World Large Claims Data to Study Opioid Use Disorder Treatment, Cascades, and Trajectory. Please welcome Dr. Qato for her talk. Thank you.
Thank you, Dr. Halabi, for that introduction. Thank you all for being here, and thank you to the Stanford UCSF CERSI for inviting me to present, and to Dr. Nathan Dang for the support. My name is Danya Qato. I'm an associate professor at the University of Maryland, Baltimore. I'm an epidemiologist, a pharmacist, and health services researcher. My talk will provide a brief overview of some of the epidemiologic challenges to using claims data to study opioid use disorder treatment patterns. Everything worthwhile is done with others, and I'm so grateful to work with my team at UMB, including clinicians, programmers, statistical support, as well as a cadre of wonderful PhD candidates who have supported this work from day one and whom I'm honored to have as mentees. I'd also like to acknowledge the team at the FDA for their collective input and support of this project. I have no conflicts of interest to declare, save that this work presented here was funded by the FDA CERSI. Briefly, I'll provide an extremely brief overview of opioid use disorder prevalence and treatment options, introduce you to our project on data-informed models to identify optimal opioid use disorder treatment trajectories. The focus of this presentation is not on the specific findings from our project as it relates to our aims, which are ongoing, but more so the focus is to discuss two specific milestones in the research project to elucidate epidemiologic challenges, that is, cohort construction and exposure definition. And then I'll end with some takeaways. So my focus is not on the destination per se, but rather the journey of undertaking this work. As many of you are aware, the US is experiencing a public health crisis of an unprecedented scale, an epidemic of opioid use disorder and related overdose deaths. It is not wholly new, but its magnitude has increased exponentially in the past decades. Over 2 million US adults are diagnosed with OUD, and over 100,000 die every year due to opioid overdose. Since 1999, over 700,000 deaths have been linked to OUD, and this is likely an underestimate. In terms of OUD care, the primary modalities for treatment, and I'm specifically talking about pharmacotherapy, have been the following three FDA-approved therapies: methadone, extended-release naltrexone, and buprenorphine, all with and without behavioral therapy. Our project is situated in this landscape of care. As the public health impact of OUD deepens and the urgency for greater access to treatment for opioid use disorder is amplified, there's a critical need to study longitudinal patterns of OUD treatment utilization, continuation, and successful health outcomes. Healthcare claims data can be a tool as we undertake these studies. The primary goal of our CERSI project was to inform the FDA's Center for Drug Evaluation and Research opioid systems modeling effort through longitudinal analyses and provision of summary-level data that can be incorporated into the model using a diversity of data sets that cut across the privately and publicly insured. I've included here the link to the FDA's modeling effort. Our project aims were to characterize longitudinal patterns of treatment modalities for OUD in a publicly insured, nationally representative sample of the Medicaid and Medicare population and a privately insured population. Our second aim was to examine the differential impact of these longitudinal patterns of treatment on critical health outcomes such as overdose and all-cause and opioid-related hospitalization. I've also included a link to our project information on the slide. The data sources for our project were Medicare and Medicaid data from CMS and commercial claims data from IBM MarketScan, now Meritive. We use retrospective longitudinal data analysis methods and descriptive methods, including visualization of treatment pathways via Sankey plots in this work. I'll start with the discussion of the cohort construction and some of the attendant challenges. In terms of the cohort definition, our data ask was limited to those beneficiaries with an OUD diagnosis claim during the study period, with the first OUD diagnosis claim of a beneficiary considered as the index state for purposes of identifying a baseline period and a follow-up period. That diagnosis claim had to have been preceded by at least 6 months with no other OUD diagnosis claim, what we determined and defined as incident disease. We also limited our original analyses to those with continuous enrollment in a health plan at least 180 days before the index date, what I'll refer to as the index period, and 90 days after. This schematic provides an overview of our study with the 180-day baseline period used to assess covariates and continuous eligibility, and later to flag prevalent versus incident OUD. I'll get to that point a little later in my presentation. And the initial 90-day post-index date period used to assess initial treatment exposure, specifically medications for OUD and/or behavioral treatment, as well as health outcomes. I want to turn now to a discussion of some of the challenges of this work, specifically with respect to the cohort construction. There are well-described challenges to utilization of claims data in epidemiologic research, namely that claims data are generated principally for billing and administrative purposes, that only services, procedures, medications adjudicated through insurance are captured, so medications, as was mentioned earlier, that are paid through cash are not captured through these claims. Furthermore, In the OUD population specifically, there are unique challenges that investigators should consider, with several populations de facto excluded due to the nature of claims data, specifically incarcerated individuals and those who are uninsured. And important sociodemographic information may not always be captured. The implications of implementing strict continuous eligibility criteria in a vulnerable clinical population on generalizability, for example, should also be considered. Inconsistency in definitions and measurement across studies is especially challenging as you undertake work using claims data for OUD and as you study and define definitions and attempt comparative analysis across research studies. Other challenges include the difficulty in ascertaining the severity of OUD using diagnostic claims alone. The ICD-10 codes have basic OUD severity codes, but the validity of these codes are unknown. One alternative approach we've used in capturing OUD severity or attempting to capture OUD severity in the baseline period is ascertaining co-occurring conditions such as hepatitis C, a medically treated overdose, or skin abscess that can be used to help identify high-risk OUD populations. The validity of OUD diagnosis claims themselves for identification of beneficiaries with OUD is also understudied. Furthermore, and finally, OUD diagnoses are notoriously underreported and undercoded due to a variety of reasons, including but not limited to provider specialties and lack of knowledge and training, challenges in diagnosing OUD, patient underreporting of symptoms, and stigma. It is because of this concern with undercoding and underdiagnosis of OUD that led to one initial decision to revisit our framing. We chose to flag beneficiaries as incident OUD if they had no claims for OUD remission and no claims for medication for OUD in the baseline period, that is the 6-month period before the OUD diagnosis index date, and flag as prevalent if they did have claims for OUD remission and/or medications for OUD in the baseline period. The Sankey diagrams I'll share here are visualizations that help us depict the pathways and flow of one treatment to another. And depicted here are the first 3 months of treatment after index date for the incident subgroup, which made up over 90% of the population sample. Notice here that the predominant therapy for the group in the first month is behavioral in darker blue, followed by no treatment in red, and then buprenorphine plus behavioral in light blue. And the details here are not important, but they're meant to illustrate a point, which I'll get to in a minute. This is the Sankey plot for the prevalent subgroup. And you notice here that the predominant treatment in the first month after index date was buprenorphine and behavioral therapy, followed by buprenorphine only, followed by behavioral therapy only. So the Sankey plots helped us visualize that the incident and prevalent subgroups were distinct subgroups of OUD. However, most studies utilize OUD diagnosis claim for index date and start a follow-up. The different Sankeys suggest that the first OUD claim is insufficiently capturing all those with OUD and thus cannot be certainly ascribed as incident diagnosis. What if we considered MOUD claims to define the cohort? What are some of the complexities in doing so? For one, the temporal relationship between alcohol use disorder, which is also treated with naltrexone, and OUD is difficult to ascertain. There's uncertainty about how to deal with behavioral health claims that are not specific to OUD, the comparability with other studies. In the absence of diagnosis, the unclear indication for buprenorphine therapy, which has other indications, including pain. And for this reason, although we began our analysis identifying the index date with the OUD diagnosis, we're conducting some exploratory analysis to examine either OUD diagnosis or MOUD treatment claims as the index date. This is indeed the recommendation offered by Johnson et al. In their piece, Making Opioid Use Disorder Cascade of Care Reality, where they say for improved accuracy, it's important to identify individuals who may not have an OUD diagnosis claim, but may have medications for OUD. Having made that decision, we wanted to get a sense of the number of beneficiaries who might be impacted. In the Medicare cohort of 868,000, the majority had no MOUD claims. And among those that did have both an OUD and MOUD claim, the majority had an OUD diagnosis before the MOUD claim. But 1/3 had an MOUD claim before the OUD diagnosis. These are beneficiaries in a cohort that are defined by OUD diagnosis that had, again, an MOUD claim before their OUD diagnosis claim. So it's possible that we are not, in fact, capturing their incident diagnosis, but that they may have been engaged in care for some time before their diagnosis claim was made. In the commercial claims data of a sample of approximately 715,000, the majority also had no MOUD claims. And among the 66,000 who had both an OUD and MOUD claim, half had an OUD diagnosis claim before the MOUD claim, and 1/3 had an MOUD claim before the OUD diagnosis, which was similar to the Medicare sample. Among those with an MOUD claim before OUD diagnosis, we also wanted to understand how many beneficiaries with what appears to be a previous OUD diagnosis would not have been captured because of our eligibility criteria. When we dig further into the subgroup, we see that the time between the MOUD date and the OUD date varied from a mean time of 9 months to a full year and a median time between 3 to 7 months. The beneficiaries with MOUD and thus potentially already diagnosed with OUD may thus be excluded depending on the continuous eligibility criteria. So we may be underestimating the significant subpopulation of the OUD population. In terms of exposure definitions, some of this was addressed earlier, so I just wanted to say that specifically for OUD, defining treatment for OUD is built on several assumptions. The remission status is unclear in claims data. Methadone is undercoded or sometimes not even covered by insurance, and behavioral services are often wrapped into the methadone. I have not touched on other modalities for treatment for OUD, including harm reduction, social and financial interventions that are not captured in claims data. And as I said earlier, buprenorphine is used also for pain. And so kind of disentangling that in the data is complex. There's also co-occurring OUD and alcohol use disorders, both indications for naltrexone. And I just wanted to plug the importance of having clinicians on your team to make assignments for day supply assignments that's necessary for understanding this data. And then finally, in terms of policy and coding changes that happen when you have data that cuts across 5, 8, 10 years, such as we had, also complicates this process. Finally, I wanted to end with some big picture points. One is that longitudinal claims data are necessary for better understanding OUD treatment pathways, transitions, and associated health outcomes. But it's important to consider the implications, limitations, and advantages of the decisions on data extraction, cohort construction, and index date definition. Sensitivity analysis can help us understand these implications, as I've shared with you today, and better understanding of the data through descriptives and visualizations can be helpful to decision-making when utilizing claims data. Again, clinician input is critical to making sense of the data and to ascertaining correct exposure assignments and days supply. The transparency around cohort construction, exposure definitions, and methods are essential, as was mentioned in an earlier presentation, for replicability of study findings and generalizability. And I just wanted to also reiterate something that was said earlier, the critical importance in investing in data linkages that better capture the totality of care needs for people with OUD, including those that are not pharmacotherapy modalities. Thank you very much.
Thank you.
Yes, another great session, so thank you. This question is for you, Molly. So one of the things about real-world evidence is that it also, and using real-world data, first of all, it's very impressive that you got that you were able to simulate an RCT or get something similar. So that was impressive. But then I wondered about can you ever think of a way that you could get pharmacokinetic data? Some kind of data that would allow a little more mechanistic or pharmacogenomic data and incorporate that into your
That's really interesting. I am not sure how I would do that. I feel like that's something that Jim should talk about. I mean, so my biggest thing, I teach a secondary data analysis class, and my biggest thing to people is you have to know your data. You have to know your data inside and out. And the second thing is no one data set can answer every question, and that includes an RCT, including the best RCT in the whole world. So, I mean, bringing together all the different kinds of data, I mean, that sounds like it's something that would be a really interesting thing that you could think about doing, and think about how you could incorporate that.
Thank you.
That was a wonderful session. And a quick question for Dr. Carter-Cal. You know, I think that's really a very exciting paradigm you describe of personalizing care. What is the next step in terms of validating it? Do you think there needs to be some experimentally validated approach to see if you can personalize based on those factors you identify?
That's a good question. This is, validation part is actually not part of this this proposal or research, but I think this is something potentially doable in the future if we do have the resource to do that. Personalize the treatment. If we understand all those confounding factors well enough, we can potentially personalize that. But for cancer, in oncology, that's going to be extremely challenging given the nature of the disease, highly heterogeneous and quickly evolving, right? That there's a time factor there as well for the cancer treatment. But I do want to learn from my knowledge standpoint to see whether other fields like cardiovascular disease area, whether anyone have ever tried to verify their their predictions of the individual levels of response. I haven't got there yet. This is a new topic to me to a certain degree. I'm still learning, but I think that's a good question.
Any other questions? Well, I have one announcement to make where if anyone is interested in submitting manuscripts regarding real-world data or real-world evidence, I'm co-editing a special issue in bioengineering, and the deadline is end of May. So if you're interested, please let me know. You all have my email. Okay, thank you. I think Kathy? Yeah.
Before I introduce the poster session, I just want to thank all the speakers today. I personally found this very informative day, and I'd like to see many more days like this where we learn a lot about the different aspects of regulatory science. I thought each of the sessions were fabulous. So, okay, now next slide. Oh yeah, I can now push it on. Okay, great. So now we're into, I guess I should also once again thank Nathan, Lawrence, Christina, Holly, all of you. Thank you for organizing today. So let's give them all a hand. And I also can't help it, Tina, I got to thank you and your entire group for everything you've done, putting this whole network together and motivating us to interact and think about themes and cross-collaboration. So just thank you so much from all of us. Okay, now to the posters. So you can see we've got 17 posters, and if I don't hurry up, we're not going to have time to even see them. And 10 are from our trainees, and they're across all the different CERSIs. And of course, they're across the range of products as we saw today, or the range of FDA centers, which I think was wonderful. And we have poster winners, and I'll screw up their names yet again, but let's try it. So Yale doesn't look as good today. I'm so sorry, but okay. So Mariam Mughali again. So congratulations. But maybe stand up if you could. There, I'm so sorry I've butchered your name, but congratulations again. Maximilian Siebert. There we go. Thank you. Abigail Abikoyi. No, I don't see her. Okay. We'll clap for her anyway. Brenda Miao. There we go. Congratulations, all of you. And then we have some honorable mentions. Trupti Shetty. Congratulations. Ruben Vargas from UCSF. Congratulations, Ruben. And Bianca Reginald. All right, so I think, Nathan, do I have anything else to tell them? The poster session is all where we were this morning, all the way down the stairs. Lower level. Okay, yeah, thanks everybody for coming. Thanks for a great day. Great 2 days.